Welcome back, folks. This is Chapter 13, Gene Regulation in Eukaryotes. A substantial amount of this chapter has been truncated, but the slides are still here. So those of you who wish to have a full understanding of this chapter may do so. But the grey slides will not be tested upon in the genetics course. In terms of content, this chapter is divided into many sections. Those which the students must learn have normal colored backgrounds, and those that are optional for you to learn will have gray backgrounds. There are some differences, major differences and minor differences, between how eukaryotes have evolved to regulate their own genes compared to prokaryotes. Some things are common. DNA is involved and proteins are involved, and the interaction of the two is paramount. So the shape of the DNA molecule and the shape of the protein molecules are very, very important. What you'll find is that eukaryotes have taken the interaction between different proteins to a new level. So we have many different types of proteins interacting at the promoter and other locations called enhancers that decide if a gene is going to be regulated. And that's fine-tuning that's not normally seen in prokaryotes. In addition, we find that RNA plays a bigger role in regulating the behavior of the end products, proteins and other RNAs, than it does in prokaryotes. Let's learn some basics first. The most important thing is that proteins are trans-acting regulatory factors. That means they can wander around the cell and bind to any piece of DNA. But once they bind to that piece of DNA, then they'll only affect the sequence to which they're bound. So in this case, they will be cis-acting from that point onwards. Let's reiterate the similarities between prokaryotes and eukaryotes. In prokaryotes, we discussed how activator proteins bind to DNA sequences and stimulate transcription. Well, the same thing happens in eukaryotes. We also learned in prokaryotes that repressor proteins bind other sequences of DNA and hinder transcription. Well, the same thing happens in eukaryotes. But the major difference is that in eukaryotes, large complexes of proteins will form around various positions on the DNA, and they then will compute how much of that gene to activate or not activate, which is not the case in bacteria. And finally, a single transcription factor, a single protein produced by its own gene, may not just control one gene product, but many thousands, and even tens of thousands, because that protein factor can be combined with others in various combinations to produce an unlimited number of outcomes. This distinction between cis-acting and trans-acting is very important for students to be able to define. Figure 13.1 may look complicated, but guess what? You already know it from previous chapters. It's just a sequence of steps going from the DNA through transcription to pre-messenger RNA, then messenger RNA processing, polyadenylation, capping, and intron removal to result in a mature messenger RNA. The mature messenger RNA is then exported through the nuclear pore into the cytoplasm, where it then interacts eventually with the ribosomes in translation, and a polypeptide is produced, and then that polypeptide goes on to fold properly and become functional. And the purpose of this slide is that at any point in this pathway, the process can be regulated, i.e. controlled. So normally, most genes are controlled at the first step, just like in prokaryotes. But in eukaryotes, we have many layers of additional control that prokaryotes don't. For instance, step number 2. Well, prokaryotes don't normally perform messenger RNA processing. They go straight through to a polypeptide. Likewise, this slide just tells us that in eukaryotes, we have many more regions along the DNA that are involved in decision making. Unlike in prokaryotes, where we found that normally the upstream proximal promoter, the promoter close to the beginning of the gene in bacteria, is enough to make the decision as to whether to copy the gene or not to copy the gene. So if you're not familiar with that, please go back to a previous chapter where we looked at lacoprons and tryptophan oprons, and rewind yourself. The equivalent region in eukaryotes is called the core promoter region, and that includes the tartar box and any other sequences that are in the vicinity to which the RNA polymerase II will bind in transcribing these messenger RNA type of genes, as well as any associated general transcription factors that we covered in a previous chapter, the GTFs. What's new are the more distant promoters. Those are called proximal elements, and they vary in number and location depending on which genes have evolved in which manner. But there are far more of those in eukaryotes than we've ever found in prokaryotes. That's a great contrast. Unfortunately for us, eukaryotes encompass a very broad range of organisms, starting at slime molds and yeasts and fungi, and going all the way up to more complicated mammals and birds and reptiles. So sometimes evolution has taken different branches, and an element may not be exactly the same in the different branches of the tree of life in eukaryotes. So this is what this figure 13.3 is trying to illustrate to us. In simple eukaryotes like yeast, the arrangement of the proximal elements that are controlling this particular gene activation site are normally located upstream, some distance. But that distance isn't huge. You can see this gap is 500 base pairs, and each one of these dots represents a possible place on the DNA that something can bind and control the activity of this gene. Looking at the bottom figure, this is in higher multicellular eukaryotes, such as yourselves. And look at the size. So this is 500, these two lines. And every time you see this dotted line hitting the DNA, you can imagine that that region has an influence in regulating the behavior of activation of gene transcription at this particular gene right here in green. And interestingly, some of these elements are not all upstream. Some of them are downstream. And some of them are even inside introns of other genes, which is amazing, as well as upstream of other genes. So this region here may not impact this transcription start site, but it will impact this one. And in some cases, we've found these elements to be on different chromosomes. So a neighboring chromosome could contain one of these regions, but those are rare. So what do we call these more distant regions? We call the one that's close to the beginning of the gene the core. How about these other ones? Well, unfortunately, they come in two flavors. Some are helpers and some are hinderers. Those that are helpers are called enhancers. They enhance the rate of transcription. And the others, the ones that hinder, are normally called silencers. So they silence or reduce the rate of transcription from that gene. And it turns out that a combination of these two mathematics, how many plus signs does this produce? How many negative signs does this produce? Well, the answer is, well, it's a mathematical answer. If this is more than this, the gene will be transcribed at that rate. If this is more than that, then the gene will be silenced to a point that it may not even be on. So in my mind, this is very similar to a computer. So the combination of all the input at that particular moment in time will determine if transcription is initiated or not, and by how much. If circumstances change within this cell and its environment, then the relationship between enhancers and silencers and core elements will change, and that will impact the rate of transcription. Just keep in mind that each one of these regions on the DNA that we've just called by a name binds proteins, these regulatory factors, right? And the interaction between the proteins bound to these different regions is key in determining the rate of transcription. Now you may be wondering, how can something this far away interact with something here? Or even something here interact with something here? Remember, DNA is flexible. It's not like a rod as depicted here in this figure. DNA can bend easily. So within a few bends, this region here can be brought to bind to this region. So keep that in mind, please. These aggregates of proteins, these coming together of multiple proteins, sometimes many tens of proteins, even hundreds of proteins, are called enhanceosomes. So there's another word for you to remember. Enhance means to enhance transcription. Some means body. So this body of proteins sitting on the DNA at various places will decide by how much to activate or deactivate gene activity. So please remember enhanceosomes. In a normal cell that's performing its normal functions, in many cases the chromosomes can overlap. So they can go into each other's territories on a limited scale and interact. That's what happens in the nucleolus. Various chromosomes, which contain genes for ribosomal RNA, send out their chromatin loops to interact within the nucleosome so that their genes can all be transcribed from a common location. So in that case, the RNA polymerase and the various general transcription factors are able to bind not just to one of the two homologs, but they can bind to the other homolog. So in that case, these proteins are trans-acting regulatory proteins. The most important thing about this new word that we just learned, enhanceosome, is that these protein aggregates can cause the DNA to bend into loops. We just spoke about that a couple of slides back. And those loops can be large or small, based on the distance between the enhancer region and the core promoter region for that particular gene. So sometimes these can be hundreds of thousands of base pairs, and in some cases even millions of base pairs wide. So the loops are going to be large. Within the enhancers and the silencer complexes, we find DNA as part of their structure. And that DNA has binding sites for proteins and for each other. So those are called the modules. So modules are binding sites that are present inside enhancers and silencers for various transcription factors. And like we said earlier, the combination of these will act like an integration circuit, like a computer. And that will then determine how much of the transcription of that particular gene or particular sets of genes is enacted. So once you start talking about this aggregation of proteins, the question then becomes, which proteins get there first and then call in other proteins? Well, the ones that get there first are called pioneer proteins because they're pioneers, just like American pioneers came to the West to first conquer the West in terms of the white man. We have pioneer proteins, and they arrive first, and they bind to these regulatory molecules on the DNA, enhancers and silencers, and then they facilitate, they help bring in additional transcription factors, and they then bring in further transcription factors. So pioneers are important, and figure 13.4 just shows us that we can have different types of pioneers coming in. So this purple molecule comes in first, and once it binds to its target sequence, then it calls in other proteins indicated here in green. Not just helper proteins, you can also have other proteins indicated in red and orange also coming into the scene. So what happens in the end is this is what the region of DNA that's controlling a single gene may look like. It'll have a bunch of activators and a bunch of repressors. Amongst those, there will be some pioneer proteins that came in initially. Interestingly, research has shown that some sequences of DNA can enhance transcription, while for other genes, they silence transcription. So the same sequence can act as an enhancer or a silencer, and that depends on which other regulatory proteins are present and bound to those regions of DNA. So this complicates our understanding of transcription even further than we thought we knew. And that takes the variability of their locations and tissue-specific regulation patterns into account, where we now predict what's going to happen in a particular series of circumstances. And this is very important for companies manufacturing drugs, because they have to work all this out in order to prevent any undue outcomes. The following two slides indicate a particular model that controls eukaryotic transcription. It was discovered first in a sequence of experiments where the scientists named it after a video game. So that's where the name may be familiar to you. But the gene itself is called Sonic Hedgehog. Sonic Hedgehog, SHH. And this gene in humans and many other mammals directs the formation of limbs. The activity of this master gene is controlled by an enhancer 1 million base pairs away from the gene itself. So this is how far these enhancers can be in relationship to the location. But as we said earlier, it doesn't matter, because the DNA can bend round, and before you know it, the enhancer is sitting next to the core promoter for SHH. Now, when this gene is turned on by a certain factor, it causes tissue-specific expression. Now, that's important for us to learn. That means in one tissue it does one thing, and in a different tissue it does something different. The same gene. How can that be the case? How can the same DNA in the same person, but in different tissues, have different outcomes? Well, let's have a look at it. So figure 13.5 shows the limb location. So this is tissue in the limbs, and this is brain tissue, neural tissue. So here we can see that the enhancer that's been used is the red enhancer. And this enhancer is not bound to by anything. So the proteins in this part of the body where the limbs are being made are only going to bind to this piece of DNA because they recognize that. Whereas in the brain, they recognize the alternative enhancer and they bind to that. So there you see a difference in the way that proteins can interact with DNA. They can bypass certain sequences depending on which combination of pioneer proteins pile up on the DNA in the first instance. So there must be something different about this location in the developing embryo compared to this location. And there is. And that difference is that all the other proteins and their genes that are being activated, remember these factors, these proteins, they are made from their own genes from somewhere. And say you have a blob of 50 factors here, 50 proteins. Each gene will have a say in producing that factor. So there may be 50 genes controlling the production of these factors, and they themselves will be activated by their own promoters and enhancers and silencers. So in this tissue here, the biochemistry may not be the same as in brain cells. Pupils don't really need to know about the next 19 slides, but I'll give you a quick rendition of what I interpret. So sometimes when you have a cluster of genes that are being controlled by a master switch, i.e. The example that the book gives is the beta-globin gene. And the beta-globin gene is not controlled independently, it has cousins along the DNA. It has, as in figure 13.6a, the gene has alternatives. So this is the epsilon beta-globin gene, this is the gamma, and this is a pseudo gene, that's the beta-globin gene, that's a delta-globin gene. So all these genes are involved in producing proteins, and those proteins are used to make red blood cells. So that's all we need to know. It turns out that there's a master control switch here that decides which genes to turn on during the development of humans. So in the fetus, some of these are turned on, in the baby, others are turned on, and then in the adult, others are turned on. And like I said, these HS1 regions that form the locus control region, they control the entire locus. Locus means this region here. And they determine which genes are going to be activated at that point in the development So what is a locus control region? It's a highly specialized enhancer. That's all it is. And we found that many genes that are produced in alternatives in the human body, for instance, the opsin genes in your eye, the ones that decide what colors you see, which proportion of red versus green, the red-green system is also controlled by a locus control region. What's been learned since is that for the beta-globin gene complex, the various locations here will bind different proteins depending on the conditions within that cell, and they will cause looping of the DNA to take place in various ways. And that looping will decide which gene is activated, which one of these downstream genes is activated. And that's tissue specific and also life cycle specific, right? And it's nothing different to what we just learned in the last few slides, that these loops are then bringing different regions, different promoters, different enhancers, and different silencers together, and then deciding which of those genes is going to be activated. Figure 13.6b shows us graphically how things change. So this is birth of a human baby. This is in the womb, and this is a neonatal after birth. And the lines represent the proportions of beta-globin detected in the blood of that individual. So you can see there are dramatic changes going on. And the locus control region is responsible for activating the right gene here at the right time. This is fantastic. We've been hawking on about these DNA loops and how the DNA bends around. We haven't shown you yet. Figure 13.7 takes the human beta-globin LCR means locus control region, and shows you how it works. So this is a general view, and this is a magnified view. And we can see the different areas, these HS regions on the DNA, how they then can bring proteins together to bind to the common activator complex. So this could be similar to what we mentioned way back here in terms of the enhanceosome. So there's the enhanceosome sitting on the beta-globin genes. These slides go on to talk about how diseases of humans can be traced back to mutations that affect the locus control region. And that has helped us identify how this system works in the first place. It goes on to say that some enhancer sequences are conserved, not just within the species, but across closely related and even more distantly related species. And those are the ones that are fascinating. And the question is, why are they conserved? Why haven't they mutated to become different between different species? And the answer seems to be that they must be very important. So by doing studies across different creatures, and by studying what regions vary and which regions do not, we get to understand the regions that don't vary much must be important. And the regions that do vary around them or between them may not be that important. So these findings have helped computers to localize areas of the DNA that are not part of a gene but are important in controlling genes. So please remember this as a genetic student. Even though less than 2% of our DNA codes for protein, there's a fair amount of our DNA controlling that 2%. And that's what this chapter is on about. If you really want to understand the nitty-gritty, you can go through the next few slides where they use the yeast as an example of a eukaryotic transcription module. So the next few slides just go into a lot more detail about how this works. And if you take your time, those of you who wish to, you should be able to read through this and figure out how this relates to exactly what we've been saying in the previous slides. For the rest of us, let's move on to the next concept. The next concept are areas or proteins that bind to each other on the DNA, and they prevent one area of DNA and its activity from influencing another region. So it's like saying one neighbor impacting another neighbor. Or if you build a wall between them, the activity of one neighbor should be more confined to their house than impacting the neighbors. And that's what we have. So we have insulator sequences. So the word sequences tells you that they exist on the DNA, but they must bind something in order to have their effect, and they do bind proteins. And these insulator sequences, they act in cis. So if a particular chromosome has bound a protein at the insulator sequence, it only affects itself and its neighboring genes. It will not affect the other chromosomes or its homologue. So a very good use of the word cis. Let's get back to these insulators. So what do the insulators do? They ensure that only the region of the DNA that's been planned to be involved in regulation is involved, and that it doesn't accidentally include other regions. Best way to understand this is to look at this figure 13.12. So in the first example, pretend that we have this enhancer sequence which has bound this set of proteins, and it has an impact on this promoter which turns on gene A. If an insulator protein is present, it may bind to its DNA sequence. If that happens in this particular cell, that enhancer will no longer be able to bend around over the insulator and find its promoter. So as far as gene A is concerned, it's going to be down transcribed or even turned off. In some cases, what may happen is the insulator, now this happens in some cases, not all, the insulator may drive this enhancer to find an alternative, less willing partner, and that will then bind to this promoter and activate that gene in that direction. So gene A is still off. In other experiments, in different situations, the presence of the insulator will then dictate which of two enhancers has access to the promoter. So if this insulator wasn't present, the normal outcome would be that this enhancer would interact with this promoter. But because it's been blocked, then the cell has an alternative, which is less likely, but now it's the only likelihood. So this enhancer will interact with the promoter and turn on the gene. It could be that this enhancer would have worked at a much higher level on this gene than this enhancer. So by the presence of the insulator, we've changed the rate at which this gene is activated. And that's a very important concept for you guys to understand. Additional findings have even more implications, and they go into some weird scenarios. So in this case, the enhancer here is driving the activation of this gene and not that gene, because it binds and brings in proteins to bind to this Tata box and therefore forming an enhanceosome. The bigger picture is things can get pretty complex, and that's the beauty of studying genetics in eukaryotes, is that they have taken gene regulation in marvelous directions. Different genes in the same cell may be regulated through any one of these protocols. The next 5 slides present some powerful data, evidence. Now, even though they've been turned gray and you won't be tested on them directly, it's important to realize that a lot of this material, especially on all 3 of these bullet points, you've come across in the past. So once upon a time, we discussed constitutive heterochromatin. That's DNA that's always on. And then we discussed facultative heterochromatin. That's DNA that can be switched between euchromatin, loosely packaged, and heterochromatin, tightly packaged. And we also, believe it or not, came across positional effect variegation in fruit flies and how they can have mottled eyes, as shown in figure 13.13. But again, that goes very deep into how we understand the effects of these locus control regions. What we really want you to learn from this chapter, one of the core learning themes, is epigenetics. Epigenetics. Epi means above, genetics means the DNA. So these are modifications, chemical modifications, normally to the DNA backbone, not the bases, but the backbone, or to the histones that the DNA is wrapped around. And those histones, or the DNA backbone, and occasionally the bases, can be modified by small chemical tags. And those tags can change the behavior by which genes are expressed. So it does belong in this chapter because we're looking at gene regulation. One of the important things to understand is that we have now discovered that these important epigenetic modifications fall into 5 very important classes. Who knows, in the future we may discover more, but so far there's 5. So the first thing is epigenetic modification patterns can alter chromatin structure. So by adding and removing small tags, you can tighten the DNA, make it into heterochromatin, or you can loosen the DNA, make it into euchromatin, or anything in between. Maybe that's how mitotic chromosomes condense? We'll come back. Number 2, these patterns are transmitted from one mother cell to her daughters. So when a cell divides, these patterns of epigenetic tags are passed on to the daughter cells. And when they divide, they pass them onto their daughter cells. The next thing is, over time, these patterns can be reversed. So that means depending on the conditions in which the cell finds itself, it may decide to make certain regions of its chromosomes less compact or more compact, and therefore affecting gene activity. Number 4, they are directly associated with gene transcription. So these tags really do have a direct impact on whether a region of the chromosome, an entire chromosome, or even a single gene is available to be transcribed. Something that's tightly packed makes it very difficult to get into if you are a protein looking for a region of DNA. We'll talk about that more in a few minutes. And the last one is they do not alter DNA sequence. So these tags are above the ACGT sequence of information. So therefore the word epi is above the genome. How does epigenetics then relate back to what this chapter spoke about at the beginning? Well, let's put it in context. The fact that nucleosomes exist, i.e. chromatin exists in eukaryotes, allows us to have another level of control that's lacking in prokaryotes. So chromatin, the association of DNA and histone proteins and non-histone proteins, is very important, and we can change that relationship to impact DNA transcription. So there are 3 basic mechanisms that have been discovered where trans-acting proteins can target DNA sequences and modify their tightness of packaging. So the first of those is that some regulatory sequences along the DNA are not tightly bound by histones. So if you have a loose association between the DNA helix and the nucleosome core, then that allows other proteins to gain access to that DNA. Therefore, those regions may be easily accessed, and therefore their promoters and enhancers and silencers may be more easily used. Let's say just that. The second mechanism is that proteins called chromatin remodelers or remodeling complexes, they can change the distribution or composition of histones, which is different to number 3, which is where you modify enzymatically the histones themselves by adding and removing some of these tags. So that's called chromatin modification. So there's a difference between chromatin remodelers and chromatin modification, and we'll look at that later. So regardless, these 3 mechanisms together can allow the cell to have different regions of its chromosomes in different states of tight wrapping, let's call it packaging is the correct word. That leads us nicely into conversations about the next few slides. Open and covered promoters. So think about it. An open promoter is somewhere along the DNA where those promoters are loosely associated with their nucleosome cores or their histone proteins. That makes them accessible and therefore more likely to be on. Covered promoters, on the other hand, are more tightly associated with their chromatin, therefore harder to access. And if you look at this range, you can have very tight packaging, less tight packaging, more loose packaging, very loose packaging. So you get this spectrum going from one side to the other of very tight packaging to very loose packaging, or even no packaging. So that's what they're saying here in this second sentence. There's a continuum, and any region of the DNA could be at any end of this normal continuum, although some regions, such as centromeres and telomeres, are very seldom found in anything other than heterochromatin, tightly packaged. So open promoters can cause genes to be expressed. But if regions are kept open, then the genes that they contain are normally found to be constitutively expressed. That means always turned on. And they do have a nucleosome depleted region. So NDR stands for nucleosome depleted region. What normally surprises students is how little of the DNA has to be depleted of nucleosomes to allow it to be revealing its promoter. Promoters don't take up much space, as we found out in a previous lecture. About 50 to 100 base pairs of DNA. So, if you look at this example here, see the distance between these two nucleosomes? And now look at the distance between these two nucleosomes. So this is a more open structure, and this is a more covered structure. So here, this transcription factor could find its promoter, or if this was an enhancer, this protein complex could find its enhancer, and indeed it could be a silencer. So that will then impact any promoters in its neighborhood and therefore affect the rate of transcription that ensues from the transcription start site. And the gene will be here wrapped around this nucleosome. And we'll see how this can then open up this nucleosome and push it out so that it can get to the DNA. But that's a story coming up in a few minutes. One surprising coincidence is whenever we find open promoters in nucleosome depleted regions, we generally do not find a Tata box associated with those genes. So there's a difference right there. But they do have a poly-A tract. So they have a region of DNA that's rich in A's and T's, but it doesn't have the consensus of a Tata region. Instead, the poly-A tract, what it seems to be doing is it attracts enhancer seeking proteins. So it attracts transcription factors that are looking for a poly-A-T tract to bind to. So once they bind, then they start binding flanking sequences, and they help position the nucleosome at the right starting point for transcription to begin. The main function of these regions is to manipulate the location of the two nucleosomes on either side. And we can see that taking place here. So this is where the poly-A tract will be in pink, and these are the proteins that bind to it. And when these proteins come in, they have a negative effect on the position of these two nucleosomes. So this nucleosome is pushed away, and it's called the -1 nucleosome because relative to this, it's upstream. And this nucleosome here is called the +1 nucleosome because it's the next one downstream. And by repositioning this nucleosome and that nucleosome, it makes the transcription start site available for RNA polymerase to come into this gap and bind there. So this is pretty amazing how nature works. In contrast, covered promoters characterize genes that are transcriptionally regulated. So transcription is blocked until the nucleosomes can be moved, that is displaced or removed altogether from the promoter. So those promoters are normally the ones that we experienced earlier. They do contain Tata boxes and other transcription binding sequences of the consensus type. So this is active competition between nucleosomes and the transcription binding factors for that binding site. This flexibility in access to the DNA at these covered sites is illustrated in this figure. So in one, we have a non-active gene. The gene promoters cannot be accessed, nor can the enhancers, because both are wrapped around nucleosomes so tightly that those proteins cannot get access to the regions of the DNA. However, in the presence of appropriate activators, these are proteins that have the authority to move the nucleosomes are rearranged. And now for the first time, there's no nucleosome on this region of the DNA, and it can now bind other activator proteins, transcription regulators, and transcription enhancers, as well as RNA polymerase. So the TARA box is available, and the +1 site of the gene, which is on the far side, is also accessible to these proteins. In summary, we've discovered that there are many mechanisms of chromatin regulation, as we went through the 5 listings earlier. So in this regard, chromatin remodeling simply refers to the modifications that reposition the nucleosomes along the DNA, so that open and closed promoters and other regulatory sequences can be made either available or non-available. And this is a summary of what we just said, that open chromatin has a relaxed association between DNA and nucleosomes, and therefore that favors regulatory proteins coming in and then activating, if conditions are appropriate, the gene. In closed chromatin, that's produced by modifications that cause the regulatory sites to be covered by nucleosomes, restricting access, and therefore turning down or turning off the genes. One of the methods that we may have spoken about earlier is a tool that was used in the early years of molecular research, and that is called DNase 1 hypersensitivity. What is DNase? It's an enzyme. What does it do? It eats DNA. So DNase 1 is an enzyme of a particular type that has been used by researchers to digest DNA into nucleotides, and it does so extremely well. Any region of the DNA that is not covered or closely associated with protein will then be accessible to DNase 1. So DNase 1 can be considered like a Pac-Man. You know, the Pac-Man game where you chase a ghost. So these Pac-Man can eat DNA and cut it to pieces if they can get access to the DNA. DNA that's completely covered and closely associated with its nucleosomes normally doesn't get cut. Any DNA that has a protein sitting on it, that protein protects that piece of DNA from getting cut. But anything in between, i.e. between one protein and a nucleosome, would then be cut. So those regions that are cut are called hypersensitive sites, because they are prone to attack by this outside agent, the enzyme, if it can see the DNA and get to the DNA. So studies like this reveal the positioning of things. So in this case, this segment of DNA would be digested by DNase 1. So will this segment of DNA up to here. But this protein may be protecting it, and therefore that part will not be digested, but this part will be. And by digesting it, it will release these nucleosomes from attachment. But the DNA wrapped around the nucleosomes will not be subject to digestion because it's too tightly packed, and the nucleosome, the DNase enzyme is too big to compete with the nucleosome. And in this scenario here, none of this DNA would be subject to DNase 1 activity. Therefore, every time we see a gene that's on, we find its promoters and enhancers and transcription start sites are normally accessible also to DNase 1. That makes sense, because then at the same time they will be accessible to other proteins that would need to bind to the enhancers and the silencers and the promoters and the transcription start site. If you have a rudimentary understanding of what DNase 1 does, try to apply that to these structures here. See if these could be digested by DNase 1 activity. Very quickly, one technique that has helped us localize the state of chromatin and to find where the enhancers and regulators and silencers, promoters are, is called CHIP technology. Now, CHIP stands for Chromatin Immunoprecipitation. So it must have something to do with antibodies and a chemical precipitation reaction and studying chromatin. And that's exactly what's going on here. So you make antibodies to your transcription factors, and those will then allow you to figure out where these are, where they're binding to the DNA, because they light up. And wherever the transcription factors are, those regions will light up, but where they're not, will not light up. And by looking at where the transcription factors are in a cell that's actively making protein from its genes, we can then discover where the enhancers and the regulators and transcription binding sites are. So this is a great tool to localize where binding is taking place. Let's look at an alternative system, Chromatin Remodelers. So chromatin remodelers are protein complexes that come in and they can move nucleosomes in three principal ways. So one type of remodeler can cause nucleosomes to slide along the DNA or to remove them from the DNA, usually uncovering enhancers and promoters. We just looked at that in the previous example. Another way that these remodelers can work is they can cause nucleosome repositioning, usually repressing transcription. So instead of moving them out of the way to reveal enhancers and promoters, they can do the reverse. They can cover them up and silence that particular gene or down transcribe it. And the third type of change is the composition of the histone octamers themselves, changing the composition of the histones chemically. So altering their interaction with each other, and therefore the structure of the nucleosome can be stabilized or destabilized. The next three slides go deep into the use of a particular example that was first discovered in yeast, but I don't think we really need to understand it to that depth, except that we now have a general understanding of how the mechanism works. Patterns are beginning to emerge as to how cells modify different histones to achieve different types of outcome when it comes to transcription and the form of chromatin that exists, euchromatin versus heterochromatin. So these modifications normally take place to the tails of the histones by adding and removing chemical tags, as we suggested. And a couple of scientists tried to work out if that pattern could be discernible, if they could figure out the code. And they call that the Histone Code. And the code would consist of combinations of chemical modifications to the N-terminal tails of histones. N-terminal means the beginning part of the polypeptide that makes the histone protein. Remember, histones are proteins, and all proteins are made of polypeptides. Anyway, interestingly, there seems to be a histone code. So more recent experiments done on Drosophila and the weed Arabidopsis suggest that of all the possibilities of the histone amino acids being modified, there seems to be four principal types that are controlling the folding or the tightness or the compactation, which is the right word, of the nuclear DNA. If this is true for other organisms, then we may have a way of regulating the chromatin structure for therapeutic purposes to treat diseases such as cancer, hypertension, diabetes, and even more dangerous diseases such as Alzheimer's and even Parkinson's. Who knows? During these studies, other outcomes were generated. One thing is that they found enzymes that would add chemical groups to the histones and to other proteins around the DNA. And they wrote these researchers called writer enzymes because they write chemistry onto chromatin. They also found another class of enzymes, and these do the opposite. They remove chemical groups from chromatin. So they call them erasers. They erase chemical tags. And then they recognize the third class of proteins, those that recognize the instructions that are written, and they read them. And by reading them, they take and perform some type of action. So writers, readers, and erases. And this seems to be an ongoing relationship along the DNA, with other proteins calling in the writers and the erasers based on their particular needs to modify the histone tails. Figure 1319 attempts to give us a graphical representation of what the readers do, what the writers do, and what the erasers do. And they also give us a relationship between euchromatin and heterochromatin with respect to the configuration of the DNA and how the change from one to the other takes place. I think the most important thing this diagram illustrates to us is that to go from loosely packed DNA, euchromatin, to heterochromatin, more tightly packed, if you add methyl groups, you will do that. If you remove methyl groups, you'll do that. And if you remove acetyl groups, you'll do that. If you want to go the other way, if you want to move the arrow that way to the left, then you have to follow the pattern indicated below. And again, a combination of methylation, demethylation, but this time acetylation drives this. So the common thing is, if you add acetyl groups, you'll go to euchromatin. If you remove those acetyl groups, you'll come back to heterochromatin. And the rate and type of methylation depends. Therefore, acetylation appears to be quite important. So let's go down and look at how that activity is performed. Further experiments have shown that inside the nucleus there are enzymes, and these enzymes are known as histone acetyltransferases, or HATs, and their job is to be called in by activators, and then when they're called in, they will add acetyl groups. This is an acetyl group, and they will add those acetyl groups to certain amino acids on the tails of histones. Since acetyl groups dissociate to become negatively charged, what you're essentially doing is you're neutralizing some of the positive charge on the histone by taking some of it away by neutralization. That reduces the interaction between the negatively charged DNA and the positively charged histone. Now it should make sense to you that if you reduce the attraction, the magnetic attraction between the DNA and the histone, therefore the DNA will be more loosely associated with the histone, therefore more loosely wrapped, i.e. euchromatin. And the opposite happens when you remove these acetyl groups, and that's done by a different enzyme. And that enzyme is called histone deacetylases, or HDACs. And these are the erasers. So there's a beautiful association between biochemistry, genetics, and cell biology. Figure 1320 is a compound figure. They try to bring in various bits and pieces that we've been teaching you all chapter into this compound drawing. You should be able to make heads or tails of what's going on in this cyclical diagram. So depending on where you are, you may have access or no access to your transcription factors. Therefore, your activity as a gene may be on or off, or somewhere in between. How about the other types of tags? Methyl groups. Well, that's called methylation. So methyl groups, there's a methyl group right there, CH3. These are added to the N-terminal tails of histones by another enzyme, a different enzyme. So these are called, well, histone methyltransferases, or HMTSs. And they are also writers. And their target is normally lysine in the tails, the N-terminal tails of the histones. And that's the one that's most frequently attacked by these enzymes, and their tags are left on this amino acid. So what does methylation do? Well, it can play a role in converting open chromatin to closed chromatin. So that's the general trend. Even though it can be used in both directions, it generally closes things up, makes them tighter, unlike acetylation, so it does the opposite. So how do you remove these tags with erasers? Well, you use a different enzyme. It's called a histone demethylase, HDMTS. and they do the opposite of the writers. So these are the erasers right there. Moving on to the next slide, what are you learning from all this? Well, there's a beautiful interplay between different chemistries, all affecting the association of DNA with its histones. So those are called chromatin-modifying enzymes. That's really all you need to know about this slide and the next. But if you want to spend a few seconds explaining things to yourself, you're more than welcome. Moving on to the next slide in the sequence, let's go and look at these pioneer factors, which combined to heterochromatin, tightly wrapped DNA, and make it available for transcription. Much research suggests that these factors may be just single proteins that have the ability to penetrate into heterochromatin and then make the necessary changes to loosen up the association between the DNA and the nucleosomes. Other research suggests that lots of individual proteins in the nucleus may come together at the right time under the right conditions to form a pioneer complex. So not a single protein, but a complex of proteins. Regardless, whatever system is at work, they will open up heterochromatin by recruiting to that location the chromatin modifiers and the chromatin remodeling complexes. Or they may alternatively bind to DNA to prep DNA for other accessory proteins to come in and form a transcription complex. Figure 1321 tries to relay that behavior. So let's go back and see if we understand more fully epigenetic heritability. So the mechanisms that are operating at the nuclear level, at the biochemical level, at the cell biology level, their job is to make sure that the chromatin is in the correct state for that particular cell type in that particular tissue at that particular point in its life, which is an amazing feat that we're still trying to discover its secrets. But regardless, we do find that different tissues in different areas have different patterns of activation along different chromosomes. So that's the secret to having the same DNA inside every cell in the body, but having those cells behave differently. And what we mean by behave differently is that they activate a different subset of genes. Some genes are the same, but other genes are different. So brain cells will produce their own set of genes as well as housekeeping genes. But liver cells will have housekeeping genes plus their own specialized genes. Thus, based on that one fact alone, you should be able to relate to the fact that different cells with the same DNA are different. Why? Well, one of the biggest reasons is the epigenetics of each cell is unique to that cell type. Once a particular cell is sent down a particular pathway and it has its pattern of epigenetics programmed onto it, it doesn't make sense that the next time it divides, its daughter cells have to start all over again. That can be dangerous. If those cells then go back to an embryonic existence, they could manufacture the wrong products and therefore ruin the tissue. So it makes sense that when a mother cell divides through mitosis, that her daughter cells continue to have the same pattern as she did before she divided. If you go back to your understanding of how nucleosomes are distributed when DNA replication takes place, so they're dismantled and half go to one newly made DNA helix and half go to the other in a random sort of fashion. This is exactly what happens with these tags. So the tags are kept on the original parental strands, and then the newly made strand simply has a new tag put opposite the tag that was on the parental strand. So the information is kept. So DNA replication is a wonderful, wonderful mechanism that has many, many layers that we are beginning to learn as students. Lastly, what's the point of having epigenetic patterns that are suited to a particular environment if you can't pass them on to your children. So in this regard, does meiosis also carry through to the sperm and eggs, these patterns? And it seems, yes, indeed it does. And we may have mentioned this to you, but it normally takes 2 to 3 generations to reprogram these genetic tags to match a new environment. Experiments with mice show that if you feed them a high methyl diet, a diet high in methyl tags, they become sick. But if you breed those mice, their children, even though they're put on a normal diet, will still be sick. And their grandchildren, the 3rd generation of mice, will also be sick. And it takes up to the 4th generation to clear those bad tags. So the takeaway message for us, folks, is this. What we eat and the lifestyle that we live is programmed into our epigenetics, and that's going to be then influencing our children and our grandchildren. So you can't say any longer, leave me alone, it's my life. Is it? Think about it. This slide just puts in perspective what we just spoke about with regard to copying DNA and separating the nucleosomes. Please read through and make sure it makes full sense to you, especially the first 3 bullet points. We spent a long time talking about DNA and protein interaction and how these proteins can modify DNA behavior either directly or indirectly. Let's turn our attention now to RNA. This first example tackles female X chromosome inactivation. In all eutherian mammals, and if you don't know what that means, you should look it up. In all eutherian mammals, the female has 2 X chromosomes and the male has 1. And regardless of how the biology works, you cannot have 2 X chromosomes or more than 1 in any cell. So what happens during early development of the female fetus? One of the X chromosomes is shut down at random in that cell. And by then you may have thousands of cells. Any cells born from that one cell in which the X chromosome is shut down will maintain that pattern forever into adulthood. So every female that we have around us is a mosaic. Every human female is a mosaic. She has patches of tissue, including the skin, that have 1 X chromosome active, and neighboring patches that have the other X chromosome active. And I think the example that came up in another chapter was tortoiseshell cats, calico cats. So they would be the equivalent. But regardless, how does that biochemical process work? We now have the answer, and it has to do with RNA. The X chromosome makes, through one gene, a type of RNA known as long non-coding RNA. So it's long because it's quite long in length compared to other types of RNA. It's non-coding because it doesn't make protein, and it's an RNA molecule. And it has an abbreviation, lincRNAs. And they play a crucial role in gene silencing in this circumstance. What happens is the X chromosome that's marked to be deactivated, it produces, through activation of a gene, XIST, so that's the gene on the X chromosome. When that gene becomes activated, it produces a lot of this long non-coding RNA, and that RNA just simply begins to coat the chromosome all the way along its length. And the result is that the chromosome becomes inactivated completely. And that pattern is then passed on all daughter cells through mitosis. Every single cell that inherits that DNA will maintain that X chromosome in an inactive form. You may remember that we call those bar bodies. So each female who has 2 X chromosomes will have a bar body in every cell in her body. Incidentally, if you have 3 X chromosome, Turner syndrome, you will have 2 bar bodies because you can only maintain 1 X to every other type of autosome. And that's the way that our biology works. Obviously, this process continues until gametes are formed. And when gametes are formed, the X chromosomes are unbar-bodied in the stem cells that produce gametes. So hopefully in the ovaries, these cells are restored back to original condition so that they can be passed on to future eggs, and those eggs won't have a bar body inside them. Here's a graphic showing that exact process at work on this particular X chromosome. So the gene lies on the long arm, the Q arm of the X chromosome in humans. And when it activates, it starts producing this XIST, which then coats the whole chromosome. And then it shuts the chromosome down by turning it into heterochromatin. Incidentally, not every single gene is shut down. There's a couple of areas that are left uncoated, and those continue to function, and they complicate our analysis of genetic disease. The next section talks about genomic imprinting. If you want to know more, you can read further, but what it basically means is that the gene that's expressed in the baby always comes from one parent and not the other. So that will complicate things. Some genes are expressed from the father, other genes are expressed from the mother. And we know now of many different outcomes. But good news is we also know the mechanisms by which they work. Let's move on to the last section. So the last section goes further and it dives deeper into these RNA molecules. A few decades ago, a new type of RNA was discovered called RNAi. And the I stands for interference. This RNA interferes with the regular process of transcription going through to translation. And it intercepts that process and downregulates it. It seems like a waste of time. Why make something only to be destroyed before you produce the protein? But it's an important high-speed regulation mechanism. So if you can intercept something near the end, you can control how long it takes to have its effect. If you're controlling something at the DNA level or even at the transcription level, it can take many hours before the effect of those changes is seen in the cell, and that may be too late. So please keep that in mind. The position along the pathway from DNA to protein and even post-protein processing is important, because the further you are along that pathway, the quicker you can respond to changes in the cell. A quick history lesson. RNAi was discovered by accident by a company that was trying to produce flowers with a deeper color, petals with a very purple color so that they could get money from Silicon Valley. And what happened is they thought that by doubling or tripling or quadrupling the gene inside these plants for producing the purple color, the petals would be very purple. What they actually saw was that there were areas of white, and some flowers were even pure white. And they couldn't explain the results until later on when they investigated the biochemical phenomenon and they discovered the RNAi. And they named it RNA interference because it was interfering with the ability of the cell to make protein. It turns out that all copies of the RNA that resembled the product that they were trying to increase by genetically engineering these plants was destroyed, including the wild type, including the normal genes. Therefore, there was no purple pigment produced, and they were then able to explain the white areas of these flowers. Since those days, we've discovered many more types of RNAi, and we've begun to compartmentalize them into specific subtypes. And you'll see that in the next few slides. But let's finish off and talk about the way this mechanism works in general. RNAi silences gene expression. That's what it does. It doesn't enhance it, as far as we know. It takes 100% of something and then it cuts it down. And it may cut it down to 0, where it abolishes any product, or it may reduce it down to 50%, 70%, 40%, whatever. And it can do that at two levels. It can do it at the transcriptional level by interfering with transcription, or it can do it at the post-transcriptional level, i.e. after the transcript has been made and it's on its way to the ribosome to be encoded. These small regulatory RNAs, and that's what they are, they're small, not the long RNA, the not long non-coding RNA that we spoke about with XIST. These are small. These are normally between 10, 20, and 30 nucleotides. These small regulatory RNAs do bind to messenger RNA in a complementary base pairing fashion. So they have a sequence that's complement of that messenger RNA that they're trying to destroy. And that can lead to the destruction of the target messenger RNA or a block in its translation. We'll see examples of that in the next few seconds. And more recently, some of these regulatory transfer RNAs have been found coming back into the nucleus and binding to the DNA to block transcription of the gene to which they can be complementary. The good news for us is that some of these pathways overlap a lot. So they may start off different at the beginning, but they merge into using the same mechanisms thereafter. They use the same proteins to achieve the same end results. Although newer discoveries are sometimes causing us to think twice. Let's begin by understanding what happens. The regulatory RNAs have to come from somewhere. In this example, they come from the cell itself. They're made by the cell. So what happens is there's double-stranded RNA produced somewhere, and we'll go back into that later. Once the double-stranded RNA is produced, it's normally a hairpin. And we've covered hairpins in other chapters, especially the last chapter about regulation of prokaryotes with messenger RNA. Regardless, double-stranded RNA is then dissected with enzymes to produce these tiny regulatory RNAs that then can go and do their job. So the double-stranded RNA made by the cell is then attacked by a protein called dicer in the cytoplasm, which cuts the double-stranded RNA into these very short sequences of double-stranded nucleic acid, about 21 to 25 base pairs. They are then picked up by a second protein, and this is called the RNA-induced silencing complex. Don't be scared, all it stands for is RISC. And what RISC does, it removes one strand of the double-stranded RNA and keeps the other one. So it digests one strand and it keeps the other one. The one that it keeps, it then affixes to itself. So it's a protein RNA complex. And it's the RNA part that's very important, because the protein then uses the RNA to go and scan messenger RNA as it enters the cytosol. Any messenger RNA that it binds to with any complementarity will then be stopped, and the protein, the RISC protein, will then begin to break that messenger RNA into pieces, essentially neutralizing it before it gets to the ribosome. So that way, this is an intercept and destroy mission by this particular protein. But the protein can be reprogrammed by carrying a different RNA. And these little RNAs can then program the protein. So this part of the protein is the same, the part that cuts, but this part here can vary. Therefore, you can have lots of these proteins moving around in the cytosol, targeting and destroying various messenger RNAs that are coming out of the nucleus. The exact mechanism of action of the RISC protein has now been determined. And what it does, it directs one of three gene silencing processes. So within the cytosol of a cell, we may have three functionally alternative ways in which RISC can be used. The first is that the complex uses complementary base pairing to attach the guide strand to messenger RNA, which is then destroyed. The RISC guide RNA binds to complementary messenger RNAs and prevents their translation. So one way is to destroy them, one way is just to bind them. And the complex directs chromatin-modifying enzymes to the nucleus where they silence transcription of selected genes. So there's 3 outcomes from the same machine depending on the end result. It's either going to destroy something, it's either going to prevent something, or it's going to change something at the chromatin level. Figure 1326 goes one step further and it introduces exogenous double-stranded RNA. That's RNA coming into the cell that's not produced by the cell. So this pathway, the cell produces its own control RNA. Here the RNA is coming from outside, a virus for instance. So both mechanisms, one is to regulate its own behavior, the other one is to protect against viral infections. They both impinge at the dicer level, and then they follow the same process of outcomes. So please study this and you'll get a better understanding of what's going on, although this isn't the best figure in the world. When you're looking at this pathway here, the endogenous pathway, we call that microRNAs. So these double-stranded RNAs are produced internally by genes by way of transcription. And once these transcribed RNAs fold back on themselves because they're palindromic, they produce double-stranded RNA. That then leaves the nucleus and is then picked up outside by various proteins. We'll see that in the next example. So in many eukaryotes now, we're discovering genes that encode just these double-stranded RNAs that are then cleaved into microRNAs. And then they go back and do a regulatory job either in the cytosol or they come back into the nucleus and control the structure of chromatin. If these RNAs are coming from outside double-stranded RNA, in this example here, we use a different classification. We don't call them microRNAs, we call them siRNAs, small interfering RNAs. And they too form a hairpin-like structure. Double-stranded RNA of a virus is by nature a double-strand RNA molecule. And when that enters the cell, and the cell happens to survive the virus attack, it will then take some of that viral DNA and use it to hunt other viral DNAs by making it compatible with the risk system. Incidentally, some eukaryotes have a RNA-dependent RNA polymerase. What does that mean? It's an enzyme that can produce double-stranded RNA from single-stranded RNA template. Viruses normally have this of the type that infect cells with RNA, but it's unusual to find eukaryotes that have this, but we've found some. The implications of this will become evident in another chapter. So some exogenous, that means outside, sources of double-stranded RNA, such as those produced by these viruses, can also trigger this mechanism of saving the cell through gene silencing. We mentioned the protein dicer, and that name is pretty appropriate since it dices up double-stranded RNA. And it has on its surface a binding site called PAS. And that PAS site is instrumental in the enzymatic activity of dicer. The size of the dicer protein determines where these PAS sites are. And if the PAS sites are on each side of this protein and the double-stranded RNA lies inside, it's going to cut at either end, and that will be about 20-21 nucleotides in length. The creation of these microRNAs and small interfering RNAs is approximately the same. What happens is that the double-stranded RNA is recognized, it's cleaved before it's sent out into the cytosol. And in the cytosol, dicer picks it up and then it trims it down and keeps one strand. And there's one strand now sitting inside the PAS site. There's a PAS site here and there's a PAS site there. And that's the active site of risk. And then this is now ready to go on the hunt. And it goes hunting to find anything that matches the strand of RNA. In the case of siRNAs, this would not be happening in the nucleus. That would be happening in the cytosol. But regardless, you're feeding into the same pathway. In animal cells, and not plant cells, there's an extra protein, drosa. Drosa sits right here. And what it does, it simply trims the ends of the primary microRNA transcript to make it into a pre-microRNA before it can leave the nucleus. There's something about these ends that prevents it from leaving the nucleus. So it produces two fragments. The first fragment is this, and the other fragment, of course, would be the black area that it throws away, and that gets recycled. So that's what we call precursor microRNA. This is our textbook trying to illustrate those processes. So there's your transcript produced by the gene, then it's cut by drosa, and then this light green is sent out into the cytosol. There, dicer cuts it here and then keeps one strand. It can keep either, but it'll keep one strand. And then that is the strand that is used to program dicer with the hunting software. So some analogy to this was made by another scientist, and he said that dicer is the hardware, the computer, but the microRNA is the software that drives the computer and tells the computer what to go and seek and destroy. You may hear one more name called Argonaut. Maybe Jason and the Argonauts, that old movie in sci-fi in the way back in the 1970s. Regardless, RISC protein itself is a multi-protein complex. It consists of many proteins coming together. And one of the proteins it contains is from the Argonaut family. So the Argonaut family of genes produces a very similar set of proteins, and they are used in many species to perform this type of function. So the Argonaut is a key component of the RISC structure. The widespread existence of this RNAi style of mechanisms in eukaryotes is thought to help prevent us from the mutational effects of transposable genetic elements, especially those that resemble retroviruses. We really didn't learn about transposable elements, but they are just selfish DNA, parasitic DNA, call it what you like. They seem to serve very little purpose for us, as far as we know, even though we're going to pick them up again in the chapters on population evolution. Regardless, for now, most transposons in eukaryotes are located in heterochromatin, and therefore they're tightly packed and unable to activate themselves. But mutations in the RNAi system can reactivate transposons by reversing the transcriptional silencing. So they can become hotspots in that particular individual or subpopulation, and that can lead to some devastating outcomes in many cases. RNAi also plays a protective role, as we said earlier, in viral infections. Indeed, in plants which can't migrate their cells, the infection of one or a few cells leads to the production of RNAi in the infected cells, and then that propagates throughout the other cells, neighboring cells, until it protects the rest of the plant from potential infection by that virus. So it's a pretty powerful thing that viruses have for them and against them. We do know now that plants that have mutations in these proteins that make dicer, such as Argonaut, they can be susceptible to the spread of viral infections. So there's the relationship between infection and the functionality of these systems. Finally, RNAi has been used by geneticists more recently as a tool. It's a tool that allows us to knock down any gene or any combination of genes and then see the effect on that organism or cell. That's a requirement or a wish list that geneticists have had for many decades, and now we have the exact tool that we need. Thank you so much.